hypertension, thrombotic precursors, behavioral changes, inflammatory biomarkers, demographic, and lastly genetics. So, the higher we expect that the higher the risk factor, then the higher the chances of getting cardiovascular disease. But that is not the case. About 60% of those who develop coronary events have only one or even none of the traditional risk factors, and more than half have either normal or mildly increased lipid value. About 20% of patients with acute cardiac event doesn't have even a single risk factor, and about 40% have one risk factor. So there is something, there are some missing links which we don't know what are those. So we are trying to find those missing links, and one of them is probably inflammation. For the HSCRP is being considered as a marker of chronic inflammation since long. There are so many acute phase reactants, but HSCRP has been selected for this purpose due to its standardization in different labs. As we all know, HSCRP is synthesized from liver hepatocytes and also from the endothelial cells. HSCRP adds prognostic information in all the level of low density lipoprotein, cholesterol, and Framingham risk score. So, what guidelines say about the measurement of HSCRP? In 2003, AHA and CDC, they reported that CRP may be helpful to direct further evaluation and therapy in primary prevention. They emphasize that they may help, but simultaneously they said that the entire adult population should not be screened for HSCRP for the purpose of cardiovascular risk assessment. But in 2010, they modified their language and they agreed that CRP in apparently healthy and elderly population to identify cardiovascular risk. But in 2013, they said that use of CRP for stratifying the high-risk patient for atherosclerotic cardiovascular disease in all age groups. This is just to refresh our knowledge of pathology days, of our third year MBBS days, how atherogenesis occurs. We all know that all stages of atherosclerosis involves inflammation, and it is a chronic inflammatory process. First step is to accumulation of LDL in intima, and monocytes leads to the intima. But recent studies have suggested that before entering the monocyte in intima, there is some population of T lymphocytes, and when LDL get oxidized, it works as a trigger for inflammation, and this oxidized LDL is being engulfed by macrophages through scavenger receptors, and they activate macrophages to release inflammatory mediators through nuclear factor kappa beta pathway, and it increases the additional molecule like VCAM and ICAM, which leads to vicious cycle of inflammation. And it also activates dendritic cell, which acts through kynurenine pathway, which we will see later that kynurenine pathway is a bit protective for us, because the metabolite of kynurenine pathway, some are harmful and some are beneficial. Like itself, kynurenine is metabolites of kynurenine pathway. We will discuss later on. And here there are these are the culprits of atherosclerosis. What are the current therapeutic options which inhibits this inflammatory process? So we have statins, which decreases the lipid level, and apart from lipid lowering effect, it has got pleiotropic effect, anti-inflammatory effect. So it checks inflammation also. We have other options options like antiplatelet agent aspirin, PPAR gamma inhibitor like thiazolidinone, and TNF inhibitor like canakinumab. Actually, canakinumab is not TNF inhibitor. It is interleukin 1 beta inhibitor. This is in phase 3 trial. So we will get result of this in recent past. So there are two novel therapeutic options we can use, like nuclear factor kappa beta. We can check it with the help of anti-inflammatory agent like curcumin, which is a polyphenol present in our Indian spice turmeric. This is the yellowish part of our Indian spice turmeric, which is being used as an anti-inflammatory agent since long in our traditional medicine. And so therapeutic options, where we stand today. So we are using current therapeutic options we have today are the statins, ACE inhibitors, ARBs, antiplatelet agents, and thiazolidinone, TNF inhibitor like canakinumab and methotrexate. These are in phase 3 trial. There is no post-marketing surveillance, or it has not reached the phase 4. Novel target are 3-hydroxyentherolic acid, that is anti-inflammatory metabolites of kynurenine pathway, and nuclear factor kappa beta anti-inflammatory like curcumin. Kynurenine pathways, there are two pathways of tryptophan metabolism. One is tryptophan serotonin pathway and another is kynurenine pathway. The end product of kynurenine pathway is NAD. And for our interest, 3-hydroxyantherolic acid is of more use. It has anti-inflammatory property. So we will discuss about this product one. 3-hydroxyantherolic acid is metabolites derived from the metabolism of tryptophan through kynurenine pathway. It inhibits activation of nuclear factor kappa beta, and its activity is to induce nitric oxide synthase, and it also inhibits antigen independent cytokines. It plays an important role in immunoregulation. So these are the study done on mice. There is no human study till date of 3-HSA effect on lipid metabolism, but yes, it significantly changes the expression of lipid metabolism related genes. It significantly decreases plasma lipid levels by 1/3 to 2/3. It decreases VLDL and increases HDL. It has anti-inflammatory effect, which has been proved by this pathological study, which significantly reduce the helper T cells in atherosclerotic lesion. It also reduces the volume of atherosclerosis. So all these studies are on mice. In summary, it is an intermediate product of kynurenine pathway, and it has clinical significant benefit in powerful inhibition of atherosclerotic lesion, beneficial effect on lipid metabolism with reduction of the VLDL triglyceride, and it increases HDL. It significantly reduces muscular inflammation. Another pathway is nuclear factor kappa beta pathway. So nuclear factor kappa beta, it remains in cytosol in inactive state as it binds with the inhibitor of the kappa beta, which after phosphorylation it activates the nuclear factor kappa beta. Any inflammatory stimuli can trigger this, but for cardiac atherosclerosis, the major factors are endothelial shear stress and activation of inhibitor of kappa beta kinase. It activates the nuclear factor kappa beta. After activation in cytosol, it translocates into nucleus and releases inflammatory mediators, and it expresses the adhesion molecules. So we have one options like curcumin, which can inhibit nuclear factor kappa beta on different pathways. So it prevents the activation of nuclear factor kappa beta by inhibiting phosphorylation of inhibitor of kappa beta kinase enzyme. It also prevents translocation of nuclear factor kappa beta into nucleus. So it prevents the release of inflammatory mediators. So it has multiple site of actions. So curcumins, it decreases the C-reactive protein and it decreases MI associated with CABG. Significantly reduces the serum cholesterol level and improves the left ventricular diastolic function. It prevents progression of diabetic nephropathy. It decreases fasting blood glucose by acting on insulin resistance. It has many other effects, like it has got anti-tumor effects. Other promising therapies are some immunomodulators are immunosuppressive promising therapies such as cyclosporine, colchicine may have beneficial effect in CAD. Methotrexate potentially cardioprotective anti-inflammatory effect through increase of adenosine level. There are 2 to 3 trials is going on low dose of methotrexate, that is 12 milligram per week in ST deletion MI cases, and one of the most important trial that is going to be published in 2017 or 18, that is CIRT trial, that will evaluate the low dose methotrexate in patients with a high prevalence of subclinical muscular inflammation. That trial is going to give an answer of all our queries that inflammatory markers are really important for atherosclerosis of cardiovascular diseases. So in conclusion, despite of acute cardiovascular events, so tackling inflammation provides the potential to improve cardiovascular outcome further. Some molecules targeting inflammation are being developed to provide a unique addition to cardiovascular armamentarium. The curcumin has a very wide range of safety, so we can use it as up to 12 grams per day.